Welcome, comrades perennials, to another episode of the audio podcast Synthetic. Artificial intelligence will not replace you, but someone using artificial intelligence definitely will.
Yeah, we are jumping right into something pretty wild today. We're looking at a major 2026 study published in Science Advances, along with several groundbreaking ArcSIF preprints.
Right. Our mission today is to totally dismantle the standard story of how life started. We usually picture the very first cell just miraculously popping into existence, like fully formed and ready to take on the world.
Yeah, the classic imagination paints this picture of a barren, ancient ocean, and then zap, lightning strikes a primordial soup, and suddenly there's a microscopic bubble swimming around.
Exactly, complete with its own internal power plant, genetic code, the ability to reproduce. But these recent sources reveal a completely different reality. The origin of life was actually this highly gradual, agonizingly slow handoff from geology to biology.
It's a huge paradigm shift because, well, we all share a single genetic code, but the transition to actually becoming free-living, independent cells that actually happen twice.
To truly grasp how wild that is, I invite you to imagine traveling 4 billion years back in time. You're standing on the floor of the Hadean Ocean. It is a completely hostile, volatile world with zero oxygen, and you're about to witness life's very first attempts at severing ties with its rocky cradle.
To understand how that eventual escape happened, we really have to look at the highly specific environment that made the origin of life physically possible in the first place.
Right, the cradle itself.
Yeah, and you are not looking at those superheated, highly acidic volcanic vents, the black smokers.
Oh, right, because those are the classic deep-sea vents people usually associate with early life.
Exactly. They look incredibly dramatic, billowing dark clouds into the ocean, but they are a massive dead end for biology. Those volcanic vents release immense amounts of toxic metals, and they operate at temperatures that they'd instantly destroy delicate prebiotic chemistry.
Like any amino acid or fragile carbon molecule trying to organize itself in a black smoker would just be annihilated by the heat and acidity.
Completely annihilated. So instead, you're looking at something modeled after a real place we can still observe today called the Lost City hydrothermal field, which is on the Atlantis Massif in the Atlantic Ocean.
The Lost City. That sounds so mysterious.
It's an area of marine alkaline hydrothermal vents, and crucially, it's not driven by active magma. Instead, it's built through this slow, steady chemical process called serpentinization.
Serpentinization.
Yeah.
That's when ocean water seeps down through cracks in the seafloor and interacts with ultramafic peridotite rocks in the Earth's mantle, right?
Yes, specifically reacting with iron-rich minerals. It causes a chemical reaction that generates heat and continuously produces hydrogen and methane gas. The fluids venting back out into the ocean are much cooler, gently bubbling up, and they're highly alkaline.
And 4 billion years ago, the surrounding global ocean was just heavily saturated with carbon dioxide, making it mildly acidic. So that creates a stark, permanent chemical contrast. And that contrast between the highly alkaline vent fluid and the acidic ancient ocean is the physical mechanism that drives everything. You can basically think of these alkaline vents as naturally occurring geological batteries.
A literal battery, yeah. Because a typical battery only functions if you have a positive side and a negative side, a physical gradient that creates a flow of energy.
Exactly. In the Hadean Ocean, the vent fluid is highly alkaline, which means it has very few freely floating protons. Meanwhile, the acidic ocean outside is absolutely loaded with protons. So you have this massive natural proton gradient.
And by the basic laws of thermodynamics, those protons are desperately trying to flow from the acidic ocean into the alkaline vent to, balance out the concentration.
It's essentially an electrical current just waiting to be tapped. But the vent isn't just an empty pipe shooting alkaline water into the sea. Over thousands of years, the chemical reactions precipitate minerals out of the water,
forming massive, towering rock chimneys made of carbonate, which is essentially the same foundational material that makes up modern coral reefs and limestone.
And the internal structures of those carbonate chimneys are intensely microporous. They are completely filled with microscopic, interconnected rocky compartments. These pores acted as the very first inorganic cell walls.
Because before life had the genetic capability to invent its own flexible lipid membranes, these tiny rock compartments physically trapped the organic building blocks.
Right, because in an open ocean, molecules simply diffuse away from each other. Entropy scatters them. But inside these pores, early molecules were trapped. They bumped into each other, solved early life's concentration problem, and began to combine.
And we can actually trace the inhabitants of these rocky pores by looking backward through the genetic history of all living things today. It points us directly toward the last universal common ancestor.
LUCA.
LUCA, yeah. For decades, the foundational assumption in evolutionary biology was that whoever lived inside those vent compartments must have already possessed all the biochemical machinery necessary to sustain itself as a functioning organism.
But the 2026 Science Advances study completely upended that assumption, didn't it?
Totally upended it. Researchers meticulously mapped the entire collection of reactions that early cells used to build the fundamental components of life. We're talking about the specific chemical steps required to assemble amino acids, RNA bases, lipids, vitamins, all from the simple hydrogen, ammonia, and carbon dioxide available in the vent.
And they mapped this essential network and found it consists of exactly 420 core metabolic reactions. But when they analyzed LECA's reconstructed genome, they found a massive void.
Right. LUCA only possessed the biological enzymes for about half of these essential reactions. The specialized proteins required to facilitate the other half of the network were entirely missing from its genetic history.
I want to stop on that concept for a second because that completely breaks our modern understanding of how a living organism functions. If LUCA is missing half the enzymes required for basic metabolism, how does it survive for even a minute?
It seems impossible.
Yeah. You look at a modern cell, and it requires all of its metabolic pathways functioning in tandem to stay alive. If you took a modern bacterium and deleted half of its core metabolic enzymes, it would instantly die.
It would just shut down.
It's like finding a blueprint for a car that includes the wheels, the chassis, the steering wheel, but completely leaves out the engine, the transmission, and the fuel lines. A vehicle like that cannot move. So the assumption has to be that LUCA wasn't an independent organism at all. If it couldn't perform its own basic chemistry, how is it categorized as life, rather than just a broken collection of half-built biological parts sitting in a rock?
Well, LUCA was incredibly functional, but you have to stop thinking of it as an independent entity. LUCA was a deeply dependent organism, inextricably linked to its geological environment.
So the missing enzymes
Weren't actually missing. They simply weren't biological yet. They were naturally occurring transition metals embedded directly in the walls of the Earth's crust. The study revealed that early biochemistry was a literal hybrid system. It was half biological proteins synthesized by LUCA and half environmental metal catalysts provided by the vent.
specifically zero-valent iron, nickel, and cobalt, lodged in the rocky walls of those alkaline vents.
Exactly. These solid-state metals naturally catalyzed the missing chemical reactions. For example, they drove the acetyl-CoA pathway, which is the most ancient biochemical pathway for fixing carbon dioxide into organic molecules.
So the environment wasn't just a physical habitat where LUCA resided. The environment was literally half of LUCA's anatomy.
Yes. The metals in the vent acted as the heavy metabolic machinery, reducing carbon dioxide with hydrogen gas to create the organic molecules LUCA needed. LUCA was essentially a biological software system running on geological hardware.
That paints an incredible picture of early life as this immobile cyborg, tethered to the vent wall like an ultimate biological ball and chain. But this introduces a massive mechanical problem.
How so?
If early metabolism relied heavily on environmental metals embedded in a rock, what was the fuel powering the biological half of the system? You run into a severe biological chicken and egg paradox right at the start of life.
Ah, right. The energy paradox.
Because modern cells run on a highly specific energy molecule called ATP. To make ATP, modern cells use an incredibly complex protein structure called ATP synthase. It's not just a chemical catalyst. It is a literal molecular motor embedded in the cell membrane.
It has a microscopic rotor that spins, powered by that exact proton gradient we talked about earlier. Protons flow through it, the rotor turns, and it mechanically churns out ATP molecules.
But here's the paradox. You need massive amounts of ATP to build complex proteins in the first place. You cannot have this massive complex protein motor without the ATP energy to assemble it, and you cannot have the ATP energy without the protein motor already existing.
It's a classic trap. But the research maps a fascinating bridge across that energetic gap, relying on two major scientific breakthroughs. First, life utilized environmental chemical energy long before it evolved the complex machinery to harvest proton gradients.
Right.
A crucial candidate for life's earliest energy source involves a reaction between phosphite and palladium. Phosphite is a reduced, highly reactive form of phosphorus found naturally in hydrothermal vents, and palladium is a potent transition metal catalyst.
So what happens when they mix?
Researchers discovered in laboratory simulations that when you react phosphite with organic compounds in the presence of palladium, you get metabolic phosphorylation reactions overnight, directly in water,
Overnight.
completely spontaneously. It requires zero ATP and zero biological enzymes. The geological environment itself was providing both the heavy metal catalysts and the chemical energy transfer reactions necessary to get early metabolism off the ground. It was forcefully pushing prebiotic chemistry forward long before ATP synthase ever existed in the timeline of life.
That answers how the raw chemical energy was available without enzymes. But it raises the question of how life actually utilized that energy to build its very first biological structures. Like, if ATP didn't exist yet, how did the first proteins get built?
Right.
This points directly to the ribosome. The ribosome is the ancient molecular machine inside every single living cell on Earth today that builds proteins by reading the genetic code.
And according to incredibly detailed research on the origin of translation from the arXiv preprint server, the ribosome does not use ATP.
Right.
Yeah, it operates almost exclusively on a completely different energy molecule called GTP.
UTP.
The ribosome is so ancient that it literally predates all known protein-coding genes, simply because it is the machine required to build those proteins. Because it is universally dependent on GTP across all domains of life, researchers can deduce that GTP was actually life's original energy currency, powering the primitive biological side of LECA while the environment handled the rest.
So the persistence of GTP in the modern ribosome is a classic example of what evolutionary biologists call a frozen accident.
Exactly. To this day, roughly 27% of a modern cell's total energy budget is entirely consumed as GTP during the process of translation, meaning the actual physical assembly of proteins.
27% is huge.
It is. ATP only became the dominant universal energy currency much later in the evolutionary timeline, specifically after the ATP synthase protein evolved and gave cells the ability to churn out practically unlimited amounts of ATP using proton gradients.
But the ribosome never adapted to this new ATP-dominated world.
No, the ribosome is so complex and so fundamentally critical to survival that any mutation attempting to switch its fuel source from GTP to ATP would be instantly fatal to the cell.
So bioenergetic evolution essentially experienced a permanent divergence.
GTP remained stubbornly dedicated to translation, basically locked in time, while the newly abundant ATP took over general cellular biosynthesis, powering muscles, neurons, and basically everything else.
The modern cell bears witness to its own 4-billion-year-old history through this strict energetic division of labor. Think about the energy your own body is using right now just to listen to this discussion. Over a quarter of it is running on an archaic energy system that dates back to the Indian Ocean.
It really is.
It is the biological equivalent of a modern trillion-dollar tech company still running its most critical servers on Windows 95, simply because the foundational code is too vital and too fragile to ever safely upgrade.
That's exactly what it is.
Now that we have this incredibly detailed picture of LUCA, powered by ancient GTP and environmental phosphite, utilizing geological iron and nickel as its missing enzymes, and living safely trapped inside the porous rock walls of an alkaline vent, we have to confront the biggest transition of all.
How do we get to the free-roaming independent cells we know today?
Right. At some point, life had to pack its bags and leave the vent. It had to figure out how to replace those stationary inorganic metal catalysts stuck to the rocks with mobile biological enzymes that it could carry around in its own membrane.
And the departure from the alkaline vents represents one of the most monumental conclusions of the recent research. As life evolved inside those rocky pores and developed the genetic capability to synthesize its own mobile protein enzymes, the transition to independence did not just happen once.
It happened twice.
Right. The two foundational domains of primitive life, bacteria and archaea, replaced those environmental metals completely independently of one another.
So they took different paths.
Researchers traced the protein structures and genetic histories of both domains and found clear cases where the ancestors of bacteria and archaea evolved structurally distinct enzymes to catalyze the exact same essential metabolic reactions.
So they were faced with the exact same geochemical problem, how to replicate the function of the vent's metal catalysts, and they found completely different biological solutions.
Furthermore, they evolved entirely different types of lipid membranes. A bacterium's cell wall is chemically distinct from an archaeon's cell wall. They built different enclosures to contain their newly synthesized biological machinery.
The sheer magnitude of that realization is just difficult to overstate. We're looking at a scenario where life didn't just crawl out of the hydrothermal vent as a single triumphant cell that went on to conquer the world.
No, not at all.
The transition from being a dependent geochemical system to a free-living, biologically independent organism happened in parallel. Both groups started with the exact same universal genetic code, the exact same ribosome running on GTP, and the exact same basic hybrid metabolism living inside the same rocky pores of the lost city.
They share the same software.
Right. But when it came time to build the biological hardware needed to replace the rock, sever the umbilical cord, and finally leave home, bacteria built one set of tools, and archaea built a completely different set of tools. We share a single, universal genetic code, but we are looking at two entirely independent origins of free-living cells.
And the parallel evolution of free-living cells leaves us with a final staggering concept to ponder, drawing on mathematical models of self-generating autocatalytic networks.
This is often referred to in systems biology as RAF theory. Right?
Yes, RAF theory. When scientists use advanced computational models to simulate how efficiently these 420 core metabolic reactions organize themselves, using only the raw environmental resources and metal catalysts available at an alkaline vent, the mathematics show that these networks form inevitably.
Inevitably.
The transition from pure geochemistry to hybrid biochemistry wasn't just a lucky fluke. It wasn't a random roll of the cosmic dice where lightning happened to strike the right puddle of mud.
It appears to be a mathematically predictable thermodynamic property of any planet that has the right mantle rocks, liquid water, and the resulting geological proton gradients.
Exactly. Given the fundamental laws of chemistry and physics, the emergence of a metabolic network like LUCAs might simply be an inevitable structural outcome of the universe itself, just as predictable as the formation of stars or the crystallization of minerals.
Subscribe to the podcast series Nuclear Locomotive, Neural Sawdust, and Synthetic. Namaste, thank you, and good luck.